Nicotinamide mononucleotide, NMN, has emerged as one of the most intriguing compounds in the field of healthy aging and metabolic renewal. At the heart of its biological activity lies its ability to serve as a precursor to NAD+, nicotinamide adenine dinucleotide, a vital coenzyme that declines steadily with age. NAD+ is deeply involved in mitochondrial function, energy metabolism, DNA repair, and cellular stress resistance. But beyond these well-documented roles, a growing body of research now connects NAD+ repletion through NMN supplementation to the activation of autophagy, a powerful self-cleansing mechanism that helps cells recycle damaged components and adapt to metabolic stress. This connection suggests a promising, dual-pathway strategy to address age-related metabolic decline by enhancing both energy efficiency and cellular renewal. As organisms age, their metabolic rate typically slows, partly due to reduced mitochondrial efficiency and increased oxidative stress. Concurrently, autophagy becomes less responsive, allowing damaged proteins and organelles to accumulate and contribute to the dysfunction seen in tissues like liver, muscle, and brain. NAD+ plays a central role in regulating many of the sensors and switches that control autophagy, particularly through its influence on sirtuins and AMPK, AMP-activated protein kinase, both of which are known to induce autophagic flux under conditions of low energy or nutrient stress. By boosting NAD+ levels, NMN indirectly revives these critical signaling pathways, helping to reinitiate autophagy and improve the cell's capacity to clear waste, maintain energy balance, and survive under metabolic pressure. Studies in aged mice have demonstrated that NMN supplementation not only restores NAD+ concentrations, but also improves mitochondrial respiration and insulin sensitivity, two factors closely tied to metabolic health and longevity. These improvements are amplified by enhanced autophagy, which reduces cellular clutter and inflammation, creating a more favorable internal environment for metabolic processes. In tissues such as skeletal muscle, which are particularly vulnerable to age-related decline, NMN has been shown to preserve endurance and reduce markers of metabolic dysfunction, effects that are partly mediated by increased autophagic recycling of mitochondria, mitophagy. This process rejuvenates the mitochondrial pool, ensuring that energy production remains efficient even as the organism grows older. Importantly, NMN-induced autophagy may also provide neuroprotective benefits. In the aging brain, accumulation of damaged proteins such as tau and beta-amyloid is a hallmark of cognitive decline and neurodegenerative disease. Enhancing autophagy through NAD+ elevation holds the potential to reduce this pathological buildup and support synaptic integrity. While human trials are still in the early stages, preliminary findings show that NMN supplementation is well tolerated and capable of raising systemic NAD+ levels, with hints of improved metabolic markers and physical performance in older adults. These early signals are encouraging and suggest that further exploration could reveal NMN's role not just in slowing decline, but in restoring some degree of youthful cellular function. One of the most promising aspects of NMN is its ability to mimic some of the cellular effects of caloric restriction, a well-established intervention for lifespan extension, without the need to drastically alter food intake. Caloric restriction naturally raises NAD+ levels and promotes autophagy, thereby increasing resilience to stress and delaying the onset of age-related disease. NMN may achieve similar benefits via a pharmacological route, offering a practical and scalable solution for those seeking to improve their metabolic resilience during aging. This makes NMN not just a supplement, but a potentially transformative tool for public health strategies targeting the aging population. While more long-term human studies are needed to fully validate these benefits, the convergence of evidence from animal models and early clinical data provides a compelling case for NMN as a candidate to support metabolic vitality through the twin levers of NAD+ restoration and autophagy activation. Rather than viewing aging as an inevitable decline, this approach supports the idea that targeted molecular interventions can reawaken dormant pathways of renewal within the body. In this context, NMN offers a window into a future where aging is not simply about extending life, but about enhancing the quality, energy, and resilience of those added years.